Hello everyone, welcome back to another session on biochemistry, and here we are going to deal with ribosomal RNAs and a few of the minor RNAs like micro RNA, small interfering RNA, small nuclear RNA, snRNA, etc. Okay, now regarding the ribosomal RNA. When we speak about ribosomal RNA, it is a part of the ribosome. It is a very integral, a very important part of a ribosome. A ribosome, it is a cytoplasmic nucleoprotein structure that acts as a machinery for the synthesis of the proteins from mRNA template. And that is the ribosome, it is formed of nucleoprotein. It is a nucleoprotein complex. In the sense, it is formed of nucleic acid as well as proteins. Nucleic acid here, it is rRNA, ribosomal RNA, and there are ribosomal protein. And these two together complex to form the ribosome, right? And this ribosome, it acts as a machinery for the synthesis of proteins. That is for the it acts as a platform where translation takes place, translation of proteins from the RNA template, mRNA template. Okay, it accounts to about 70 to 80% of the total cellular RNA, and it shows a very high degree of secondary coiling, complementary intramolecular and intermolecular base pairing by hydrogen bonds. Okay, what happens is the rRNA, it is actually single stranded, as you can see over here, starting from over the 5' end over here. If you just trace back the whole structure, it is just a single stranded structure ending in 3' end over here. Okay, but what happens is the this what you call the rRNA, the secondary structure for rRNA, it is usually, this is a secondary structure, it is usually formed of the double-stranded stems and single-stranded loops or bubbles, and this is happens because of the localized coiling and base pairing, and it is simply because of the complementarity. Okay, complementary sequences which are found within the what you call the strand, the rRNA strand, it brings about the uh, localized coiling and base pairing. Okay, we can see that the hydroxyl, the rRNA, it is a ribonucleic acid which contains a ribose as the pentose sugar, right? So the hydroxyl group on the 2' carbon of the ribose sugar in RNA, it prevents the formation of a double helix, stable double helix. Okay, so what happens is it can only lead to the formation of a like such kind of a secondary structure where the secondary structure of the rRNA, it is a complex pattern of short double-stranded stems which is interspersed with unpaired single-stranded loops and bubbles, and this is what we can see over here. Okay, so these red shaded parts, it is the double-stranded what you call stems. We call it as a stem. You can see here stem, right? All these red shaded parts. On the other hand, these plain parts, okay, these are single stranded or unpaired because they don't have any complementary sequences over here. Okay, so these are known as loops or bubbles. All these are unpaired parts. Okay, so here you can see that because of the complementary sequences on these two strands, it has taken up the intramolecular base pairing. Okay, but because of the and it has resulted in the formation of a stem. But on the other hand, over here it is still unpaired single stranded loops or bubbles. So this is the what you call the secondary structure of ribosomal RNA. Coming to the different classes of ribosomal RNA, we can find the ribosomal RNA in various groups or various kinds of cells. In prokaryotic cells, we can see the prokaryotic ribosome. Uh, the prokaryotic ribosome, it is of what you call the 70S type. Here the S represents the sedimentation. Okay, it represents the sedimentation coefficient. S represents the Swedberg unit. Okay, the Swedberg unit, this one, and the 70S, it represents the sedimentation coefficient of the prokaryotic ribosome. So what is sedimentation coefficient? It is the rate of sedimentation, the time taken for sedimentation in the centrifugation process. So when a cell content, when the cell constituents are subjected to centrifugation, how much time, what is it, the rate of the sedimentation is represented in the particular unit, 70S in the case of the prokaryotic ribosome, and in the case of eukaryotic ribosome, it is 80S. Okay, any kind of ribosome, it is formed of two subunits, a larger subunit and a smaller subunit, right? So the prokaryotic ribosome, it is the complete ribosome, it is 70S, because the molecular weight is only 2 lakh. You can see here, it is this much of molecular weight, while the eukaryotic ribosome, again of larger and smaller subunit constitution, it is a molecular weight, it is actually like 4.2 into 10 raised to 6. Okay, so this is the two kinds of ribosomes, very common one. Now, it is based upon the molecular weight and sedimentation coefficient only. All these like divisions are being done. Now, when you consider the 70S prokaryotic ribosome, it is formed of two subunits, as already mentioned. The larger subunit, it is 50S type, while the smaller subunit is of 30S type. Okay, the 50S larger subunit is further formed of two different kinds of ribosomal RNAs or rRNAs, and almost about 34 ribosomal proteins. The two different kinds of ribosomal RNAs are the 5S rRNA, which is composed of only 120 nucleotides, while the second type is 23S rRNA, which is formed of 2,900 nucleotides. Okay, so these are the two kinds of ribosomal RNAs which complex with 34 ribosomal proteins to form the 50S subunit of the prokaryotic ribosome. On the other hand, the 30S subunit, the smaller subunit of ribosome, the prokaryotic ribosome, is formed of a single ribosomal RNA and 21 ribosomal proteins. rRNA, it is 16S type, which is formed of only 1,540 nucleotides. Okay, it is, the length is just 1,540 bases. So these here itself, in the case of 70S ribosome itself, we can see it is formed of three different kinds of rRNAs: 5S, 16S, and 23S. Okay, coming to the eukaryotic ribosome, including the human ones, it is 80S type ribosome, and it is formed of two subunits, the 60S and 40S. The 60S larger subunit, it is formed of three different kinds of rRNAs and almost about 49 ribosomal proteins. Of the three kinds, you can see it is 5S rRNA, which is formed of 120 nucleotides, 5.8S rRNA of 160 nucleotide length, and 28S rRNA of 4,700 700 nucleotide length. Okay, so these three ribosomal rRNAs form the what you call complex with the ribosomal proteins to develop the constitute the 16S larger subunit of the eukaryotic ribosome. Coming to the smaller subunit, the 40S smaller subunit, it is formed of, again here also you can see that the smaller subunit is formed of only a single rRNA complex with 33 ribosomal proteins in the case of 40S. Okay, and the single type of rRNA, it is 18S, formed of 1,900 nucleotides. So these are the different kinds of rRNAs found in the prokaryotic and eukaryotic cells. And we can even find different kinds of this rRNAs in the case of vertebrate mitochondrial ribosome, etc. If you go through different kinds, we can find different kinds of ribosomes. Okay, but these are the major ones. Okay, we have prokaryotic cell and eukaryotic cell, and we have respective to that we have prokaryotic ribosome and eukaryotic ribosome. Okay, so I hope this is clear. Passing on to the next, minor RNAs. Minor RNAs, it is the first one is, it is a small nuclear RNA. The small nuclear RNA, it is a subset of small RNAs. That is, it is a type of minor RNA. And what is the function? A small nuclear RNA, as the name itself, it suggests it is present inside the nucleus. And what is the role? It do have a very important role in the processing of rRNA and mRNA. Okay, when we discussed on the post-transcriptional processing of mRNA, there actually these snRNAs do have a very important role. Okay, the whole process we haven't discussed, just what are the different kinds of processing happens in the hnRNA to mature to form mature mRNA that we discussed in the one of the previous presentations. Here we can see that the small nuclear RNA play a very important role in the rRNA processing and gene regulation in the nucleus, as well as in the post-transcriptional processing of mRNA, that is intron removal of the introns, and the processing of mRNA precursors. Okay, and that is done with the help of the different snRNAs, which is named U1, U2, U4, U5, and U6 snRNAs. Okay, these usually get complexed with nuclear proteins. Okay, and they are known as snRNPs, ribonucleoproteins we call. Okay, so those are involved in the intron removal. Okay, that is a removal of the intervening sequences and like splicing together of the exons. So there it plays a very important role. Small nuclear RNA. Okay. Involved in mRNA and RNA processing. Okay, now coming to the next two groups, the microRNA and small interfering RNA. These two, these are microRNAs as well as the small interfering RNAs. They are involved in inhibition of gene expression of certain genes by decreasing the specific protein production. Okay, so it actually interferes with the protein production, and this is done by inhibition of gene expression. Okay, so how does it do it? As the name suggests, microRNAs, they are small in length. It is almost like 21 to 25 nucleotides in length. Mature microRNAs are of 21 to 25 nucleotides in length, but this is usually formed, this small microRNAs are usually formed from precursor microRNAs, which are single stranded, but the length ranges from 500 to 1000 bases. So during the processing, the such a large precursor miRNAs, it will undergo processing to develop into the 21 to 25 nucleotide length microRNAs, mature ones. Okay, now these mature RNAs, how does it do it? Usually hybridized with the mRNA. Okay, at certain points, at certain positions. So what happens? We get a microRNA and mRNA duplexes. mRNA, they are single stranded, right? So wherever that like this microRNA, it complementarily binds with the mRNA, what happens is a duplex is formed, and usually this duplex is formed at the 3' untranslated region of the specific target mRNA. Okay, what happens as a result? The translation will not be completed. So translation, it get stopped in between, it get arrested. Okay, as a result, that particular mRNA, it will not get translated, and hence that protein, the protein which was supposed to be produced will not be produced as a result. Okay, the protein production, the specific protein production is completely getting arrested. I hope it is clear. So microRNAs, they are single stranded. Okay, the mature microRNAs, they are highly specific. They hybridize with the specific complementary sequences on the target mRNA. So this target mRNA, when the microRNA it binds to, at that particular region, duplex is formed. Rest of the mRNA is still single stranded. Okay, now the duplex formation, it will result in translation errors because the translation process cannot be completed. The protein production is like stopped in between, it is blocked in between. Okay, so that is how the protein production is reduced. Okay, now the small interfering RNAs, again, as we have already mentioned, that also inhibit the gene expression. How is it done? It is at the protein production stage, or it is at the translation process stage. Okay, so it interferes with the gene expression by decreasing the protein production, that is by actually targeting the translation process. Both miRNA as well as this siRNA works in the same way. Okay, these siRNAs, they are derived like from double-stranded RNAs, almost like about these are also 21 to 25 nucleotide length, and usually they form the siRNAs also hybridize with mRNA at the like on the target mRNA, specifically on the target mRNA. And what is the difference? It can actually bind with or hybridize at any length within the length of the mRNA, anywhere within the length of the mRNA, wherever the complementary sequence exists. miRNA, we specified that it is usually towards the 3' untranslated region, but in the case of siRNA, it can form hybridization, or it can form hybrids with their distinct targets potentially anywhere within the length of the mRNA. Okay, wherever the complementary sequence exists, the complementary to the siRNA, wherever there is a complementary sequence to the siRNA can bind to the mRNA. And this RNA, that is siRNA and the mRNA duplex, it results in the blocking of the protein synthesis process. Okay, so it is the protein synthesis process is actually abundant in between. Okay, so that particular target proteins are not produced. Okay, so this is actually done by siRNAs and mRNAs. Okay, fine. Thank you.